Proceed. So thank you for joining me today. My name is Dr. Adarsh Kumar, and I used to work as a postdoctoral fellow at Florida State University, USA. Now, next week, I'm moving to Germany to start work with Structural Genomics Consortium. Today, we'll be discussing a little bit about protein structures and visualization, and what can we learn, or what if you're looking for something with structure, how do we do it? So the question arises is, why should we study the protein structures in the first place? So because we should study them because biomolecules are nature's robots. They pretty much work to keep us alive, and because they are able to perform their functions like catalysis, gene expression, communications, transport, etc., by coiling into specific three-dimensional structures. And proteins have applications in industries that can be exploited as drug targets. So the whole structural bioinformatics relies on the holy trinity of sequence, structure, and function. So a sequence is basically highly mobile. It doesn't have a definite shape, and it is pretty much inactive. When this sequence folds into a unique, precisely ordered shape, it becomes stable and active. These structures, these active structures, then have form special associations with other moieties like other proteins, nucleic acids, small molecules, ions, etc., and which impart a specific function to this particular structure. Now, let's have a look at the motivations behind learning the structural bioinformatics. So, the first one would be the structure determines the function. So, understanding the structure is necessary for understanding the function. So, this field provides invaluable structural context for mechanistic analysis, leading to better understanding of function. Then, structure is more conserved than the sequence. So, if you go through several related protein structures, and protein structures are especially which form the same function, you would observe that sequence may be vastly diverse, but the structure will be conserved according to the function. So evolutionarily, even more distant proteins, they can share a common structure if they are to form a similar task. Then the structure is encoded in the sequence. It's something similar to the second point. The slight difference being that even though sequence can be vastly different for similar structures, the structures are still a function of the sequence. And then I'll discuss a little bit about these ones with an example which I came across while working. So the next question would be, what are the goals of studying structural bioinformatics? The first one would be analysis. That is, we do it because we want to analyze some structural data, like how conserved a particular protein's domains residues are. Or we want to visualize the three-dimensional arrangements of a protein or its interactions in the active site. Or we want to compare, how similar or dissimilar two proteins are. Or we may want to predict a structure. If we don't know a structure for a protein, we can use the sequence similarities or ab initio methods for prediction. Or if we want to design something, design a protein with a specific form and function in mind, we can pretty much do all of these. And in recent years, the advancement of computational technology has given us substantial amount of power. And if we are creative enough, we can virtually address any challenge that is thrown upon us. Applying those goals, these are some of the major research areas that people focus on related to this topic. So it could be protein structural classification, structure prediction from sequence, binding site detection, binding prediction and drug design, modeling molecular motions, predicting physical properties like stability, binding affinities, design of structure and functions in itself, or designing synthetic proteins, or improve industrial design. Moving on to the primary focus of today's discussion, that is protein structures. We'll start with a question of the what. I mean, what exactly is a protein structure? So the short answer is they are a three-dimensional arrangement of peptides, which give it a definite shape and function. But a protein isn't merely that. It's not just an amino acid and simply a chain. So to understand this, we have to begin with a very basic building block, like from the amino acid itself, and then we have to understand the level of organizations, and then which in turn gives a fully functional protein. So this is one slide just to show the existing amino acids. You can see the side chains in particular are highlighted. So it's basically a NH2CCOH. So that's your amino and acid group, and the side chains are responsible for giving it specific properties. So, for example, these are hydrophobic. These are hydrophilic amino acids with basic amino acids, acetic amino acids, and polar amino acids, or the hydrophobic ones, or special amino acids like cysteine, glycines, and proline. So to impart a particular structure, a particular function to, or a particular shape to a protein, the side chains are the ones which are primarily responsible. Then the key forces that are present, like when the protein folds, there are certain four forces which are responsible for this, their structure and even their function. So, which I'm going to discuss now. Let's discuss one by one. Starting with the hydrogen bonding, it's pretty common phenomena. Highly electronegative atom like oxygen or nitrogen or fluorine has a hydrogen attached, which gives it a partial positive charge, and then this partial positive charge interacts with another highly electronegative atom, which has a partial negative charge and forms a hydrogen bond. So what you see here is the case of water molecules, and similar bonds can be found in proteins as well. The next one would be van der Waals, which is basically a distance dependent. It is a distance dependent interaction between atoms and molecules. So they are not. They do not result from any chemical or electric interactions or electronic interactions. So they are very, very weak. And for these forces to act, the atoms need to be very close to each other. So usually these forces are involved in protein interaction with other molecules or other surfaces. The next one would be electrostatic interaction. These are ionic interactions. So it's not the delta positive, delta negative. It's actually a proper negative charge and a full positive charge. And this is how, for example, this is the interaction between lysine and glutamic acid. So they can form electrostatic interactions. At the same time, they can also form a hydrogen bonding. So you can see it in the second image. Then the next one is hydrophobicity. So hydrophobicity is a Greek origin word which means water curing. Hydro means water and phobic means curing. So name is self-evident. They try to repel water. So what they tend to do is when you put it in an aqueous environment, they try to stay away from it and try to go into the core. So these are quite strong interactions, and you can even find them in insoluble proteins, but usually they are present towards the core. If they are on the surface, most likely the protein interacts with lipid membranes. So, for example, what you see here, these are transmembrane domains, and they will be formed of hydrophobic amino acids. Then the next one is disulfide bridges. So it is a covalent bond, so it is one of the stronger ones. It forms between two cysteine, the sulfur sulfhydryl groups of two cysteine, but they are susceptible to reducing agents. So if there is an oxidizing environment present, they will form a sulfur-sulfur covalent bond. If there's a strong reducing agent in the environment, then the bond will break and they will have free sulfide. So moving on, amino acid sequence, they just don't abruptly go into any 3D coiling form, coiling and form of functional structure. So there are levels of organization, or even higher, we can call them hierarchy, and we're going to discuss a little bit about that. So let's take the structure organization first. So starting with the primary structure, so it's just amino acid sequence, linear, coiled, whatever, but they don't have any definite fixed structure. Then these primary structures fold into secondary structures. Secondary structures could be alpha helices, which I'm showing right here. These are helical forms. Usually N and N plus 4 amino acid residues are form a hydrogen bond, and they will have, and it's usually a right-handed. It is a right-handed bond. It is a right-handed. Then the other secondary structures could be like there are other helices as well. For example, 310 helix, where N and N plus 3 hydrogen, N and N plus 3 residues form hydrogen bonding. So they are like, for example, if your alpha helix is this wide, then the N and N plus 3 would be slightly narrower because the that it's quite close. And then there is pi helix where N and N plus 5, which is wider. So pi helix existence is questionable at best, but 310 helices are found in about 10 to 15% of all helices in the protein structures. And for example, it is also found in myoglobin. The second one would be beta sheets, where it consists of beta strands. They are flat strands, and they are connected laterally by at least 2 or 3 backbones, forming usually they are generally twisted and pleated sheets. And they can be anti-parallel, where the one strand goes one way and the other another way, or they can be parallel, where both the strands go in the same way. You can see this is a porine protein from PDB ID 1A0S as an example. Now, while looking at these hierarchy, the next thing we can see is the motif. The motif is basically a short conserved sequence pattern associated with a distinct function. So, for example, this is a zinc finger motif, which is 10 to 20 amino acids long. Usually, motifs are, they are part of a functional domain or part of a protein. They cannot function by them, and that is the point which differentiates motif from domain. Domain is also a conserved sequence pattern, but they can function as an independent unit. So they are usually larger, so like for up to 700 amino acids, and they may or may not include motifs within the context. So, for example, these are transmembrane domains. So even if I chop off this area and this area, they can, like this domain can still interact with the lipid bilayer. So domains are independent. Domains can function as an independent unit. Next one, the next hierarchical step is tertiary structures. That is the fully folded protein. It will have all the domains, motifs, or even the secondary structures in it. And this is the final functional form. And beyond the tertiary structure, you can have quaternary structures. This is hemoglobin, where more than one tertiary structures interact with each other and form a bigger form of function. Now, come to the how part. That is, how to determine the structures. So we can do it either experimentally or computationally. So experimentally, we can use crystallography, whether X-ray or neutron crystallography. There are electron lasers as well. Then small angle X-ray scattering or cryo-electron microscopy. And computationally, we can go for structure predictions, which is either homology modeling or de novo structure prediction. So nowadays, you would hear about artificial intelligence-based structure predictions as well. So if you're following the news for past few years, then the most recent cast, they had DeepMind AI, which is supported by Google, and they did a pretty good job in predicting almost every protein that is known. Now, the PDB. Now we discuss a little bit about the protein data bank, that is PDB. So PDB is where all the experimentally determined structures are submitted. So now that we have a general idea about the protein structures, let's have a look at this server and determine what information we can take it from this repository. So we'll come to the file format. So to access it, you can either search Pdb directly, or you can go to our CSV Pdb. This is the home page, pretty straightforward home page. That's an overview of structural biology. There you'll have a molecule of the month, some features and highlights, latest entries, and then you will have the data of Pdb, how many structures, how many from human, how many nucleic acid structures, and all this. One link that I would suggest, if you are interested in learning about it, you can go to the Learn section, and you can find everything which is needed to understand the structures or browsing the Pdb data bank. But for now, we'll focus on the search bar. Question is, what if you want to look for a particular protein? So, for example, I used to work on a protein called human DNA polymerase beta. So, search, just write DNA. This gives you lots of results, like really too much of them. Now, the next step would be that we want to refine the details. So if you have any special chemical attributes, or if you want to search even a sequence, you can go through these options. Else, the refinement options are on the left side. So I want to see from Homo sapiens. That's eukaryote pretty much. So, for example, you don't want just Homo sapiens. You want the whole whole eukaryote. You can uncheck this, and then you can check the eukaryote. So I want from Homo sapiens. I want extra diffraction structures. Let's say release date, I want the latest ones. It's transferase live, so that's fine. All alpha protein. If you are interested in something based on the structural, content, you can go through scope classification as well. Right now, I want to do this, so click next, and you have all the structures which were submitted recently. 6 in KR and KS. Now, I am biased towards these entries because they are mine. So, ternary complex 1, ternary complex 2, 26. Let's So this is one of the structures that we submitted. So when we open the entry, you will have tabs on the top. We'll come to them in a moment. You have how the structure looks as a biological assembly. If there are any other biological assembly, you will have a biological assembly too. What is present in the asymmetric unit. And you can directly visualize from here structures, electron density, and whatnot. And you can have a look at the details. So for example, classification is transferase, DNA transferase. Organism is Homo sapiens and synthetic construct. So Homo sapiens because the source organism for this protein is Homo sapiens, human. And synthetic construct is given for the DNA, the experiment. But we did express it in E. coli, so that's the expression system. It is a wild type, so there are no mutations. Date of deposition and release. This is the experimental data snapshot. Most of the time, you'll see this will tell you how good the resolution is and how well the data is stored. That's the publication record, and then you have all the related structures as a list. When you go down, you will have macromolecule details. So that's DNA polymerase beta, which is chain A. That's the protein part. And Homo sapiens, all the details. And there are nucleic acids. So you click on nucleic acids. There are 3 chains. So that's entry 2, 3, and 4. You will see everything here. Then there are details of small molecule ligands. So DCTP, that's a QPJ, that's an artificial one. Then pyrophosphate and magnesium ion. Then again, the experimental data and validation is a slightly detailed report. And then there is the entry, history, and funding. coming all the way up on the 3D view. So for example, if you don't have any software with you, and let's say you're going somewhere and you want to just have a quick look, you can directly visualize the protein on the web page itself. So that's that. I just use this tab to visualize sometimes when I'm working on my phone or something, and when I'm traveling. But most of the time, I use the softwares. For example, Primal, which I'm going to show you. Then annotations. All the related annotations for all the chains are present here. Experimental details would have the details of crystallography. So if you're interested in replicating the work, you can check this tab. Sequence will have all the sequence details. Genome information. It will have all the chemical, everything available, hydrophobicity, any disorder present, and genome variables. Then for genome, since we have, while submitting the data, we give it with what is the sequence and what is my source. It automatically correlates it with the genome entry in NCBI or so, and then it gives us a proper location where it is present on the genome. Then there are ligands. What ligands are present in this one? So there's QPJ, one of the ligands, and there are others as well. But here. Then that's the pyrophosphate, DCTP, and like. And if there are any versions or revisions, you will have the versions. So for example, 1.0 was initial release, and when the database was cross-referenced and citation added, that's the second version. Now, if you want to download a particular protein structure, you should go to the download files options. It will always be present all across the tab. So if you want to download the sequence, that's your sequence. Then Pdb format. Let's say you want to download the Pdb format, you click on the Pdb format and just download it here. And you can open it with Pymol. Now, before we proceed, I would like to discuss a little bit about the Pdb file format. So Pdb file is not very complex. It's not a very complex file. It is a text file basically. So the software reads that text file and converts it into the structure which you can read. So you can actually open the file and read it. I'll show you a number. So it not only lists the coordinates, it lists everything which I showed you in different tabs, more or less. So for example, it will have a header which will have title and author, several remark columns. I'll show you in a moment, and then atom and heteroatom are the atoms are for the protein and DNA. Heteroatoms are for water and small molecules and ligands, and they denote where each and every atom is located. So so here, this is one of the related files, that is 7RBI. You can just right click, open it with Notepad, and you will have all the details. So for example, you start with the header. Header says it is transferase DNA, and this is the date of deposition, 6 July 2021. And that's the PDB ID 7RBI. Title, whatever title was given by us. Compounds, that means which molecules are present. So that's polymerase beta, then which is one, which is engineered. Then you have DNA, and everything. Same thing, organism expression system, which we just discussed. That's the author and title and everything. Resolution refinement, all the experimental details. Again, same thing, experimental details, extra diffraction method, and the completeness and everything. Then coming down, you will have some missing atoms. There are missing residues and there are missing atoms. So missing residues will tell you which complete residues are missing. So for example, 9th amino acid, let's say it's a valine, and valine is missing altogether. So it will be listed at this point. So in this case, 9 is glutamate. So glutamate is missing. Missing atoms means the residue is present. So for example, some of the side chains are quite mobile. So let's say one of the arginines are quite mobile. It has a long side chain. So sometimes we are not able to model the whole side chain. So we just delete side chain and leave the basic form in the chain. So in that case, it will tell you which atoms are missing. So for example, in the 10th amino acid, which is threonine, atoms gamma oxygen 1 and gamma carbon 2, they are missing. So in 48 lysine, these two atoms, and so on. Then you have singles coordination. This is the whole sequence, right from here all the way to the end, with along with the 6 histidine tags. This is the location of histidine tag, 336 to 341. Then you have the DNA sequence. Then what are the heteroatoms present? So heteroatoms mean whatever the ligands. So you have the QPJ, pyrophosphate, magnesium, DCTP, two molecules. This will show you the helix and sheet location, like from which amino acid to which amino acid your helices are present at, and where the sheets are present. So one thing, if you know how to code and if you know how to write a program, then it's easy to understand. It's easy to find out all the details from this particular file. So these help you as an identifier all the life, and they have a definite format, so you can actually let's say count the number of spaces and so on, and then you try to use it to extract the data. Then you have link records. This is for it will show you a little bit of flashes or okay. Now coming to the most important. So whatever is present till this point above, even if I delete the whole thing, like the whole thing from here all the way up to the top, I delete it, save it, the file will still be readable. Okay, it will still be readable by the software, because the most important part is this one. These are the coordinates. So for example, atom is designated as atom. That means it would be either for protein or the DNA. That's the first atom, which is a nitrogen, which is designated as N, that is nitrogen, of threonine, which is present in chain A, and that's the 10th residue. This is the designation. This is the element symbol. That's nitrogen, and you have values 1, 2, and 3. These three values are Miller synthesis, which is you can simpler terms. Uh, you can assume it as X, Y, Z in a three-dimensional space, and it will tell where exactly that atom is present. The 1 represents the probability of that atom being present at that space. So this is called occupancy. For example, if the probability, if this value is, let's say, 0.5, that means that atom is present in 0.5% of the molecules which cause the diffraction. So not 0.5, sorry, 50% of the molecules that cause the diffraction. And this value is called Wilson's temperature factor. It tells you how much vibration is present. So the higher the value, that means the vibration is a lot. So let's go down. So same thing again. Let's take this one. Atom, 67th atom. That's the C delta 1, which is from leucine, 19th amino acid. Same thing, 1 occupancy, and the carbon. Ca is not calcium. That is C alpha. Cd is C delta. Cg is C gamma. Cb is C beta. So similarly, OE is O epsilon. Now let's see if we have some partial occupancy. I know we have some partial. Anyways, before going to the partial occupancies, these are the detail. These are the residues for the DNA. So that's why it is DG and DT, DA, DC. If it is RNA, it won't have D. It will just have A, U, G, and C. Then go down. Now comes heteroatoms. Okay, you see there's a TER card, TER. The TER card tells the program that that is the termination point for the chain. So there should be a TER in here as well. You see, between the chains P and T, there is a TER. Similarly, there will be TER on the top side. Now coming to heteroatoms. Heteroatoms, these are ligands which we present. So this is QPJ, that's the ligand. Chain A401, same thing, it is fully. All of these occupancies are 1, but have a look at this one. That's the pyrophosphate, PPV. This has an occupancy of 0.3. That means when we diffracted the crystals, this pyrophosphate was present at 30% of that. Similarly, this one has an occupancy of 70%. So what we were trying to do in this case, so that you will have an idea why we got different partial occupancies, is we try to capture the reaction in process. So for example, you cannot actually, because these are crystals, you cannot actually capture the reaction in process. But when let's say half of the reactions are done, half of the reactions are done, or 30% of the reaction is completed, you will have that broken pyrophosphate in 30% of the reaction, and 30% of reaction will have the product. But the rest of the 70% will have the reactant in particular. So that is this 70%. Then HOH is for water, all the water molecules that is present here. Then these are connect records for binding. These are needed for software to read this particular file. Now coming back to the PPT task, I would like to request you all to take some like about 5 minutes and try to do a little bit of search by yourself. And if you have any questions, you can directly ask, or you can put it in the chat box for the time. So I wrote it that search AP endonuclease. It's not mandatory. Search whatever you like, and try to go through just the information so that you see to the next. Okay, we have a question here that for myosin 6, human option is not 6. It's right here, Homo sapiens. Myosin 6, Homo sapiens. Okay. Yes, those three numbers are coordinates. That's a good question. Can we add missing atoms? They won't be experimentally correct if you want to add. There are softwares where you can build atoms. So hold on to that thought. I will discuss it when I discuss PyMod. No, you shouldn't. So you should not add if it is not present in the experimental atoms. If you want to do some sort of simulations, for example, you can just add that because simulation will take care of the orientation and everything. They will take care of the physics and chemistry. But otherwise, just for observation, you should not. If the atoms are missing, that means those atoms are highly mobile and you cannot capture them in one or relatively similar position all across the structure. So we have a question: what does ANISOU means? ANISOU. So some of the protein structures are not very straightforward to solve. So and in that case, you uh, do a refinement that is called TLS refinement. What that refinement does is it will kind of shake each atom slightly and uh, over the XYZ axis, and it will try to fit it as the best way as possible. In those cases of refinement, these NISO records come into existence. You have to add those records to the file before. For docking studies, are those missing atoms needed? It depends. It depends. So, for example, let's say if you let's say you have a side chain. Let's say you have a arginine side chain, and that is highly mobile in the structure. But that is actually needed for something to bind to it. So, if it is you know if it is bound to a certain ligand, it will basically be at a fixed position. So, it's better if you generate those side chains. Like if you look hard enough, you may be able to find examples where the apoform of the protein structure has some side chains missing from the active site even, and when it is bound to certain ligands, those side chains are back in the place. So that's one of the things. Anyways, so let's proceed with PyMol. So PyMol is freeware. It's not exactly freeware. It's an open source software. It used to be a freeware when it was with Delano Scientific. Delano is a developer of PyMol, but now it is owned by Schrodinger. So you can download it. You can download it for free. Use it for 15 days trial, and after that, it will ask for uh, license. But if you are using Linux, Ubuntu or CentOS, whatever, you can get an older version which is freely available at, I think it's available at GitHub. You'll have to check. So for Ubuntu, you can directly download it, like sudo apt-get install PyMol, and you will get it. But if you are using Windows, in that case, you'll have to download and you can use it even without license, but there are certain limitations, and I'll talk to you about it. Okay. Before we proceed to the PyMol software in particular, with the PyMol usage, this is the resource, the main resource actually, PyMol Wiki. Any question that you have on how to use PyMol, you will find that question in here. So you can use PyMol with as a you can use commands in PyMol, or you can go with fully with the graphic user interface, and you will go all the answers in this. So let's discuss PyMol. So let's open the 7RBI. It's the same file which I just showed you as a text. So double click it. Right. My PyMol has expired in my Windows PC, so I'll just go to skip activation. Even if it does even if you skip activation, all the functionalities are more or less similar, except for this tagline, both license files, so for evaluation. And then if you try to generate a figure, let's say, you will have a watermark like this on your figure. So it's not exactly usable for generating figures in particular, but for visualizing and everything, you will have all the details, like all the functionalities more or less, at least so far that we commonly use. So on the top, you have the usual functions, file, edit, build. If you want to build something, just like as someone asked me if you can add crystals, you can, even in PyMol, but I use, there's another software which is much better in this regard, that is Coot, COOT. Then you can create a movie. I've never done that. Usually, if you want to create for some presentation or something, that's a good option. Then these are displays, settings. Edit all is something you would use quite often. So for example, if I have to set cartoon, this representation is called cartoon. So if I have to set cartoon color from this option, you can choose color. That's color. So that is default. We can change it to, let's say, red, and that becomes red. So I think, yes, the Rasmol is free to use. It's been a while I have used Rasmol. PyMol is the more potent of the software. So whatever uh, figures that you see in the publications, uh, most of them, as far as I know, more than 90% of them are made in PyMol. So for me, it's just a matter of convenience. So there are other softwares as well. So if you want to explore, there is Chimera, one of the softwares which we can explore for visualization. That is also free. Chimera is free. If you want, you can get Discovery Studio Visualizer. Um, that is also free to use, but you'll have to kind of register and get a download. So PyMol, I'm sharing PyMol because this is one of the most common ones for publications. which you can find in pretty much every lab. Then on the bottom, you can see the mouse mode. Mouse mode will tell you in what mode the mouse is. So currently it is in 3-button viewing mode. So whatever I'm doing, it's for viewing. And the best part is the combination. So rotate, move. So it's left, middle, and right mouse button and wheel. So it's rotate, move, and move Z axis. Okay. So that's the Z axis, like go in and out, so zooming in basically. And if you try to make a combination with shift, control and shift, and single click, double click, all the details. If you want to change it to editing mode, just click once here and it changes to editing mode. And then your options also change slightly and you can see. Mostly we'll be doing viewing, so I'm just keeping it at viewing for now. Now you can see on the bottom there are movie controls. Since I don't have any movie file here, so I cannot go much into detail, but one of the options that is S stands for sequence. So when you click S, it will give you the whole sequence of whatever present in this file and which is on your desktop. In this panel, you have A, S, H, L, and C. So A is actions, S is show, H is hide, L is label, and C is color. So for example, so this is one of the structures. If I want to say zoom in, orient, and let's say I want to align. Usually, well, this is one of the common ones which you'll use, but I don't have anything else to align. So I'll come to this option. Then for show, so at this point, everything is in cartoon representation. What I want to do is I want to see the sticks. So I'll do sticks, and there you have sticks as well. But I don't like the cartoon interfering with the sticks, so I'll go to hide, and I'll hide the cartoon. There you go. So that's all the stick representation. There are several other representations as well. So, for example, one of the ones which you would have seen in some figures is surface. It will take some time. Yeah, so that is a surface. You can clearly see in this one. Then there is a mesh as well. So these are the visualizations. So what I did at this time was that I simply opened the structure from the file. But now what I want to do is I want to download another structure from the file. So what I'll do is I'll go to plugin, this option, get PDB. I'll go to get PDB. I want to find another PDB from another file from the PDB database. So this is 7RBI. Let's say I will add 7RBI. When I download it, that's your 7RBK. I'll change the color, let's say, to green, just so that. Wait. So you can see these two are two different colors. Change this color to slightly. So that's yellow. Okay. Right now they look pretty much overlapped. because of the positioning. But if they weren't, you could use the action key to align the molecules 7RBI, and now it is well over. So these two are essentially the same uh, uh, molecules with slight differences in the reaction stages, so they are pretty much aligned to each other. Just for the sake of it, and that's the alignment. Sometimes you want to do alignment because you want to see the movement of something. So, for example, let's get the 7RP. 7RP. I'll hide this one. Action 9, 7, okay. It is aligned for the most part. Okay. But if you look at this region, see that there is a movement. So during the course of reaction, from binding all the way to product formation, there is this movement between binary and ternary form. This is something you can judge when you have two structures and when you like this. I'm showing you just an example of why you would be using uh, comparison alignment for comparison. Then okay. Next would be what I showed you here was just overall alignment, like the whole protein to whole protein. There are options where you want to align to a definite area of the of a protein. So I'll fill that up. So this is a protein which I worked on during my PhD. You can see it is coil helix with a zinc ribbon domain on the center. Now there's another protein. This one. That's also a coil helix with zinc ribbon. So remember in one of the slides I was talking about structures being more conserved. So this is one of a good example. These sequences are about 12 or 15% identical. Like even less than 30% identical. But they are still, they look largely the same. If you overlap them, if I want to overlap them for any study, for example, I would, let's say, molecule 506. That's the alignment. But it is not exactly helping. That's not like you can't get any information other than that it's a whole uh, jumbled hodgepodge of two different proteins. But leaving the N terminal, this coil aside, as I said, both of them have C terminal zinc ribbon. So the point is, are they very superimposable? So what I will do is I'm going to select the C terminal region. So it starts from 176. So I selected from the sequence, and this is the 5Y06. You can see here that's the 5Y06. So I'm going to rename it selection 5, just for okay. Then I'll do the same thing with this one. Again, rename the selection. Okay. What happens when we align these two selections? So I'll check this one, or let's say I'll align actions and align. Now I'm not going to align to molecule, I'm going to align to selection. So align one selection with the other and have it. This region is very, very well aligned. Zinc ions for both of them, all the loops, helices. So you can find a good example of helix. Both have parallel and anti-parallel helices. So let's switch off one. This strand and this strand are going in the same direction. So that is parallel helix, and this strand and this strand is going anti-parallel direction. So that's anti-parallel. So this is now when we made a selection, we can look at this. We can clearly look at this one. So what I can do is I can hide everything. Let's say I just want to look at the C terminal because the N terminal is distracting. So I can hide everything. Then I can go to the selection and show it cartoon. Select this one, show it as cartoon. You would observe that the atom is missing, the zinc ion is missing. And the reason is you can't represent a free ion of the cartoon representation. So what we can do is here, select this zinc ion and this zinc, show them squares. For them squares. And there are. So this was just an example for selecting a particular region if you want to analyze that particular region. Then another thing, a very simple basic thing. So let's hide a bit. Now, for example, if you want to understand how far something is, or distance between any two, let's go to right. So, for example, I want to see how far these two atoms. They're not relevant in terms of distance, but just for example. So I right click this one, and let's say double right click this one. It gives me a distance of 4.3 angstroms. If I want to save it, I can type in the box DIST. That's the distance. Save it. That distance is saved. So even if I uncheck it, that distance is. If I want to measure angles, for example, I want to measure, let's say, what is this angle? I can double click here, here, here. That gives me an angle of 114.3. Similarly, if I want to check the dihedral, for example, so I can that's minus 64.4. So you can see it will also show you the dihedral. So if you are into if you want to do such an analysis, that is also possible. So okay, as for by these are the basics ones. You can do PyMol. You can use PyMol for pretty much a lot of things, but most of the things that you'll be coming across are these. Now, before we end the session, I want to show you one of the figures that, like, as an example of what you can do with it. Look at. So what I have shown here is that these are electron densities for these particular nucleotides. So that's DCTP, and these are the part of the DNA which we saw while we were exploring the Pdb structures. This is just to show you what sort of capabilities Pine has other than just visualizing. Then, if you want, let's say if you want to save this figure. So what I do is this is just an image. You can save this as an image, but it will have all the white background and everything. But if you want to prepare in terms of figures, you should have a ray traced image, clear background. So what I do is I'm going to make it ray trace. So I type it, focus on this region on the PyMod. So you have a command line here and the command line at the bottom. You can use either of them. So ray tracing, so ray. If you have a good computer, you can proceed to like 2000. That's 2000 pixels by whatever the height is. That gives you a 300 dpi roughly. But in my case, my computer is not very powerful. So just one example, I'll make it. When you enter, it will take some time. And it generates a ray traced figure. Ray tracing will have everything, like it will have background, it will have, it won't have background, it will have shadows, a proper lighting and everything, and you can use this figure. You can export this figure actually. So for exporting as PNG format, you can type PNG space, let's say, it says row 1000 by 434. So I traced it as ray 1000. So the width is 1000, and whatever the height is, so that's 434 pixels to the file here. If we go, I should have that file. Now, since my license file is not present in this case, you will have something like this. This is not a high resolution file, so it's not usable for figures. But yeah, that is one of the examples I wanted to share. For figures, you want to go to ray 2000 or ray 3000, depending on how big your figure, how big are the figure size. That's one example. Proceed towards. So yeah, so we are pretty much done with the demonstration. If you have any questions, you can ask me now, and then we'll submit it to class.
Sir, from these protein structures by PyMol, what we really have to predict?
PyMol is basically a visualization and analysis software. It's not exactly prediction. Okay.
Okay.
So I'll give an example. Now, for example, the structure that I showed you is from this particular.
Okay.
So when I did the experiments, I solved the structures, worked on the biochemistry and everything. But once the structures are solved, I have to analyze them, right? I have to look at them and to get to any conclusion. So for example, you see all these figures, how
Okay.
the distances change, okay, between different stages, okay. Then
Okay.
how the binding coordination changes. For example, calcium has a 6 coordination number, so the calcium is present, and sodium has a variable number. Magnesium has 6 numbers, so that is present. And so on. Then
Okay.
these are different stages of incorporation. So I was talking about the uh, occupancy. So in this case, it was 30%, 50%, 70%, and then the PPI is 100%. So that's the completion of the reaction. And I have not included the binary stage here because binary doesn't have the DCTP, but binary will have this helix n in open position, like just like this one. So that's open, and so it was open, then it closed, and then it went open again. Okay.
Okay.
Similarly, this is one of the this is the second active site, where I can see how well things are presented. So Pymol was used to, just make these figures and to study how the protein was behaving in different structures, all through the structures. That was basically. If you want to do prediction, let's say if you want to predict a protein structure, then you'll have to go to prediction softwares in particular. So Discovery Studio has a good prediction software. Schrodinger has a good prediction software. Or you can just go online and you can go for SysPDB server. And then I think DeepMind server, the AI is also present. Then
Okay.
Rosetta Fold is there. Okay. So there are
Okay.
several.
Okay, sir. Thank you.
Anyone else?